All right, we're going to pick up where we left off and head back toward cardiac looping. We're also going to discuss some of the vessels, although we are going to cover that in more detail in the next set of lectures. But as sort of a basic introduction to the blood vessels of the heart, because of course the heart would not be the heart without its vessels, we start with a pair of dorsal aortae. So you have one major aorta coming out of your heart, but initially you have a paired set of aortae that are that sort of develop from bands of mesenchyme that run parallel to the notochord. Okay. From those dorsal aortae, we get the aortic arch arteries. Now, during what's something called convergence, we get during the craniotocaudal folding. Oops, sorry, move backwards instead of forwards. Okay, so in this process of convergence, during the cranial and caudal folding, so we've got the front fold and the back fold essentially, they're bringing what's called the atrial pole, which is really the outflow tract, okay, together with the venous pole, okay, which is the sinus venosus, or the other end of the heart. And that brings those two things together. And that kind of looping of the heart is what helps to position all of the chambers in their proper positions, as we sort of ended on the last time. But it also puts the vessels in their proper positions. Again, we're going to cover vessels in more detail later. We're going to do sort of a quick trip through it now. Okay, so early development of the vessels. In week 4, those two paired dorsal aortae fuse and they become the single aorta. Okay. Also during week 4, the heart starts beating. We talked about the heart being the first organ to be functional in the embryo, and circulation of blood throughout the body begins. Okay. We also see fairly significant remodeling of the venous system. So these are your veins. These are the vessels that are carrying blood back toward the heart. Now, initially you have a lot more veins than you're ultimately going to have, and many of them are going to regress. But in looking around the region of the heart, a lot of those early veins are going to disappear or regress, and one of them, in particular, is going to enlarge, and that's going to become the superior caval vein, also called the vena cava. There's also the inferior caval vein, okay, down there, also, or the inferior vena cava, okay, that's one of the major vessels of the heart. The left sinus horn, and when they're talking about sinus horns, it's a point of convergence for all the vessels, okay, in this case it's for veins. So from the left sinus horn, so an area where all of these veins are converging, it becomes the coronary sinus. And what the coronary sinus is a vessel that receives blood from the coronary veins and transports them into the right atrium. Okay, so it runs along sort of that curve of the heart. Finally, the pulmonary vein, now that in this case we're going from one vein to several. Okay, so the pulmonary vein starts out as a single vein, but ultimately it's going to sort of diverge into 4 separate veins, producing 4 separate openings, allowing blood to move between the left atrium and the lungs. And those pulmonary veins are actually going to grow up toward the lungs and merge with the vessels in the lungs to complete that circulatory pattern or pathway. Okay, but again, a lot more on vessels in later lectures. Okay, so septation, valvulogenesis. We have to be able to make our 4 separate chambers, and we have to be able to create valves that keep blood flowing in the right direction. So how do we make septa? There are a number of different ways that we can do it. One way is through differential growth and remodeling. So essentially we have structures that are growing at different rates, and because they're growing at different rates, their growth patterns can affect the structures next to them. So for example, if you have one structure that's growing very quickly next to a structure that's not growing as quickly, it might impinge upon the smaller structure and restrict its growth. And that could be a normal pattern of growth and development. So it's that kind of give and take that can produce septation between, or in an empty space within a developing organ. We also see some structures that regress and disappear. So you initially are forming structures you ultimately won't need. That's certainly one way to do it, and that's part of it. But another way, and something that's critical in the formation of the heart is the formation of what are called cushion tissues. And the way that that happens is you start with the myocardium, which is going to secrete some extracellular matrix in between the myocardium and the endocardium, so in between these two layers of the heart. And producing that extracellular matrix essentially underneath the endocardium sort of forces it to expand into the lumen, or the space of the heart, creates these bubbles on the walls of the heart. Now, then some of the endocardial cells will convert to mesenchyme, which will then enter that area of extracellular matrix and become connective tissue, which is called cushion tissue. And that's shown there. Additional mesenchymal cells and neural cross cells are also going to populate the cushion tissue. So we have other cell types that come in and use that extracellular matrix as a framework and sort of grow and develop in the region. And then finally, as the cushion tissues are growing, they will merge. So the cushion tissues tend to be paired on opposite sides of a lumen. And as they grow together and merge, they form a septum. And that's another way to do it. Two major areas where we see cushion tissues forming to produce septum are the outflow tract and the AV canal. Okay, and like we talked about, AV canal defects tend to go along with Down syndrome. Another term for the group of heart defects that tend to go along with Down syndrome are endocardial cushion defects, which makes sense because they're failure of the cushion tissues to develop properly. Okay, so next up, forming a septum between the two atria and dividing the atrioventricular canal. So in this step of heart development, we're essentially taking what was a tube and making 3 chambers out of it. Ultimately, you're going to need 4. Humans have a 4-chambered heart, but we're going to start with 3. Okay, and those 3 chambers will be 2 atria and a ventricle. And the way that we start is with a myocardial or a muscular septum called the septum primum, which forms in between, at the midline of the single atrium. Okay, so in between what will be the left and right atrium, but at the midline of the single atrium. And then it sort of elongates from the cranial dorsal wall. Okay, essentially the top and back of the atria. And that's a differential growth process. So we've created this septum. It's growing sort of out into the heart. Along the sort of leading edge of the septum primum, you get this proliferation of dorsal of mesenchyme, okay, and it's called the dorsal mesenchymal cap. So we've got that. Additionally, also dorsal mesenchyme, we have the dorsal mesenchymal protrusion. So this is another growing structure that runs in between the dorsal mesenchymal cap and the dorsal AV canal cushion. Okay, so we've got all these different structures all growing in the same region towards each other. So the dorsal and ventral AV cushions, or endocardial cushions, are going to fuse. So this is a cross-section of the AV canal. And what we're seeing here is that the dorsal endocardial cushion and the ventral endocardial cushion are growing together, ultimately forming a septum, separating the outflow tract into the 2 separate vessels. Okay, so we have right and left atrioventricular canals. Also, during this process, the dorsal mesenchymal cap, the dorsal mesenchymal protrusion, and the atrioventricular septum, okay, as they're all sort of growing and developing, are going to fuse together, dividing the single atrium into the left and right atria, okay? And that whole process of all these structures sort of growing together and fusing gives us 3 chambers in our heart, okay? You can see in this picture the small ventricular septum that's starting to grow. We're going to cover that later, but initially you have 2 atria and a ventricle. All right. So, a really, really interesting structure that's part of the atrial septum, the foramen ovale. Okay, this is actually a hole, and it's not just any hole, it's like a tiered hole that runs between the 2 atria, and it allows blood to flow from the right atrium into the left atrium, which would allow deoxygenated and oxygenated blood to mix, which normally we don't want. But in a fetus, we do want it. Because remember, a fetus is not breathing air, so blood traveling to the lungs is not picking up oxygen. So the normal patterns of circulation that work for anybody who is born and breathing aren't going to work for a baby in the womb. The oxygenated blood comes from the umbilical cord. And when we talk about vessels, we will talk about how that alternate structure works. But this foramen ovale is part of the system that helps keep oxygenated blood running to fetal tissues as they're developing, when the baby is essentially growing a circulatory system that won't work for it until it's born. Okay, so the foramen ovale. Essentially what we've got here, as I said, is a tiered hole. So in, the atrial septum has got sort of two separate sections, okay? And there are holes in each. Now, what happens then is that the blood can flow in from the right atrium in through the first hole, and then sort of into the space between the two septa, okay, the septum primum and the septum secundum, okay, and then it flows out through the hole in the septum primum and into the left atrium, okay. Now, the septum being a two-piece arrangement here is critical because the septum secundum is very thick, not exactly rigid, it's muscle, but it's a little bit thicker and a little bit stiffer, whereas the septum primum is very thin and essentially floppy. So blood that flows towards the septum secundum can pass through it and into the space between the two septa and then out through the hole in the septum primum. However, blood in the left atrium that would try to flow back, just because the septum primum is so floppy, just pushes it back against the septum secundum, and it can't get into the space and then out through the hole into the right atrium. So it's only flowing one way, okay? After birth, changes in blood pressure force the two septa together and they fuse so that essentially the hole is gone, okay? The two original holes are still there, although I'm sure they grow together such that they don't exist anymore, but that immediate change in blood pressure forces them together so that you can no longer shunt blood between the two atria. And that allows for the changes that are necessary for breathing air as opposed to getting your oxygenated blood through an umbilical cord. There are some children who are born with what's called a patent foramen ovale, or PFO. Essentially, a foramen ovale stays open after they're born. It's not usually something that requires correction, although it can. It's not immediately life-threatening, but ultimately would need to be fixed if it doesn't go away on its own. But there are babies with certain heart defects who actually benefit from having a patent foramen ovale, because for certain heart defects where blood isn't circulating properly anyway, having a shunt between the right and left atria may be